Okay, hello everybody, and this is our recorded class this week. And what I'm going to do is I'm going to finish off the section that we were discussing DNA and DNA function. And then I'm going to move on to our next section, which is microbial metabolism. The two are, of course, intimately linked because what we're talking about in DNA function is how DNA actually drives the functions of the cell through the products that for which it has the recipes in the genes. So first of all, we ended up talking a little bit about genetic manipulation and genetic engineering, biotechnology, etc. So I just want to recap the most important points about how we produce recombinant DNA. And that is DNA from two different sources, which are put together, manipulated and put together for some particular purpose. And that is the simplest definition of recombinant DNA. So here are the basic principles. What we're usually going to do is we're going to make use of a vector to move DNA around, to move genes of interest from one source to another. Now, those vectors most commonly are bacterial plasmids. The plasmids are replicating circlets of DNA, like very small chromosomes, and they often code for very useful products and properties like virulence or resistance to antibiotics, heavy metals. But they have some other properties which make them particularly attractive for biotechnology, as we'll see. So here, for example, is a plasmid, a euplasmid, which is useful to us of one sort or another. Many have been characterized and many have been constructed to use. Here it is in its host bacterium. We grow up a whole lot of the host bacterium in culture. We lyse them and we extract and purify the plasmid. Here it is here. And once we have those plasmids in solution in vitro, then we can actually start to manipulate them. Then we look actually for a source of DNA which is going to be useful to us. We are going to just use the example of human growth hormone, in which we're going to extract from the human genome. Now, that gene only exists in one place on the human genome. The human genome is enormous. So we have to know how to find that gene. That is the product of years and years of research. But once we know its sequence and we know the sequence of everything that is on either side of it, we can cut it out of the human genome and end up with bits which just consist of the gene itself, the gene for human growth hormone. And what we're going to do is we're going to take that single gene and we're going to insert it into the plasmid. This here is the plasmid there. The plasmids actually can be quite large, but it's simplified here. Here is our gene sitting in the circle of the plasmid there. Now what we have to do is we now have to take the plasmid and we have to reinsert it. This is why it's called a vector. It is a vector because it's shuttling DNA into a living system again. So we take that plasmid. It is now a recombinant plasmid. It consists of a bacterial plasmid and human DNA. So it's a recombinant plasmid. And we insert that back into bacteria. And if everything's gone well, then the bacterium grows up. We get billions, trillions of bacteria growing, and they actually start to read off the product of this gene. And in this case, is human growth hormone. We can then, once that has happened, once we're growing all our bacteria, we can actually extract the human growth hormone, which is now a product of this recombinant DNA in the bacterial cell. So I'll just mention that many genes have been moved into plasmids in this fashion for various uses. And in this case, we're using bacterial cell, But you can actually also use plant cells, depending on what you want to do, and even animal cells if they are being grown in culture. So all of these yeasts, by the way, are also used. Single-cell fungi are also used. And the end result is that we commandeer the functions of a normal cell, and we get it to do things which are useful to us. We can modify many, many different organisms in this fashion. And remember that one of the reasons for that is that DNA, by and large, operates the same in any organism. All organisms, their life is based on DNA, and the basic functions of DNA are the same. There are differences that have to be accounted for, but in the end, this has been extremely successful. Here, for example, is growth hormone actually being used to treat somebody who suffers a growth hormone deficiency. Okay, so let me just say that we have skipped over something which is tremendously important, and that is understanding how we cut out that gene and how we insert it. How do we get it to stick into the plasmid? Well, it depends upon a neat trick and an incredible feature of bacteria, of microorganisms in general. And that is that microorganisms have their own type of immune system. Bacteria are particularly susceptible to virus infection and have many mechanisms. We'll hear about another one at the very end of this. They have many mechanisms to recognize the presence of pathogenic virus and to attack its genetic material. In this case, what we're particularly interested in is the way in which it attacks the DNA of DNA viruses. There are also RNA viruses, and that's another story. But we're interested in the DNA viruses because the way in which the microorganisms attack the viral DNA makes it very useful to us. So the reason for that is that they use a set of enzymes called the restriction enzymes. And the restriction enzymes, there are many of them, but there are some which are of particular use to us. And we're going to look especially at this one. I'll just mention. This one is the one that we're particularly interested in. They have specific names depending on their characteristics. This is BAM-H1. It's extracted from a bacillus. This is ECHO-R1. It is extracted from E. coli. Now, the restriction enzymes act like a little pair of molecular scissors. They recognize specific, very short sequences in the viral DNA, and they go along and they cut it, and they inactivate the DNA by doing that. It is what they recognize and how they cut it that makes it particularly useful to us, because the restriction enzymes recognize a specific sequence on the genome of the virus or on a piece of DNA. They will recognize the sequence, whether the DNA is extracted from the virus, whether it's extracted from humans, whether it's extracted from plants. It doesn't matter. What they recognize is sequence, is DNA sequence. In the case of BAM-H1, this is what it recognizes, GGATCC. And there is something very peculiar about this little short sequence, and that is that it is palindromic. It reads the same. On one strand, it reads the same as it reads back on the other strand. So we go GGATCC. Here we go GGATCC. And look how it's pair bonding. It's going GC, GC, AT, TA, CG, CG. So it's a palindrome. It's GGATCC, GGATCC. And the restriction enzyme cuts between these two Gs. It cuts there and it cuts there. Now, the one that we'll pay particular attention to is this one, ECHO-R1, and it reads GAATTC. and it's a palindrome, so it reads the same backwards, GAA-TTC, and the base pairing is G to C, A to T, A to T, T to A, T to A, C to G. And again, they cut at between the G and the A, the G and the A. Now, the effect of this is that those pieces of DNA separate from one another. They split apart. That is how the restriction enzyme inactivates the virus, cuts up its DNA. We give it a big piece of DNA to cut at one specific site. It will open up the DNA and it will leave these hanging ends there and there. They will actually separate, and we call those sticky ends because you'll see they could stick together again. If I bring if I've got a sticky end that's reading if it's reading TTAA like this, I can bring in another piece of DNA that reads AATT, and it'll base pair there and stick to it. Now when we use this in recombinant DNA, we make sure that those things don't join up easily until we want them to join up. There are ways of ensuring that they stay apart. But the effect is that we split the DNA, we cut the DNA, and we end up with these sticky ends that are ready to base pair with a matching palindrome. So let's have a look here. This is actually a very famous plasmid called PUC19, which is very frequently used in bioengineering. And here, for example, you see here's the BAMH1. There's a BAMH1 site there. There's another one called Hindi3. But here's the one that we are particularly interested in, and that is Echo R1. So, no, hold on. I don't want you. I don't want you. I want you back. Here's the Echo R1 site. So you can just picture in your mind that there is that little there is this cuts area here. And if we cut here, we're going to end up with two sticky ends. We're going to open up our plasmid and there'll be two sticky ends there. And that is very useful because that means we could, in fact, insert a piece of DNA cut from somewhere else. So this is what is actually done. Here is our source DNA. This is let's say, for our example, this is the human genome. And we know that the human growth hormone gene is here. We know that because we've done a decade of work on the human growth hormone, and we've sequenced the DNA. And we know that here's the human growth hormone. And on either side of the human growth hormone, there is an Echo R1 site. That means a place where Echo R1 can cut. So we come along and we incubate human DNA with Echo R1, and we end up with these cuts. Here they are. Here's the Echo R1 site, GAA-TTC, GAA-TTC. Here's another one on the other side of the gene. Here's the gene here. Here's another one on the other side, GAA-TTC, GAA-TTC. And they cut here, there, and they cut here, there. And what do we end up with when that happens? We end up with sticky ends reading AATT, and here on this piece, AATT. And we're going to now take this piece and we're going to treat it really carefully, purify it, try and ensure that it doesn't flip back on or join or degrade or whatever. And we're meantime going to go to our plasmid, to our bacterial plasmid, PUC19. And we're going to cut the PUC19. We only have to cut it in one place. Then you go back here. Picture, if we cut just at one Echo R1 site, what are we going to end up with? We're going to end up again with two sticky ends. And because it's Echo R1, they're going to read AATT. So we're going to cut. Here they are. Here it is here, AATT. And we're going to join them in. And we got one, we'll have one hanging, one sticky end on the one strand, one sticky end on the other, and our insert will fit in and it will stick, it will base pair. So here it is here. Here's our source DNA from human, AATT. Here's our plasmid, AATT. So they base pair there, A to T, A to T, T to A, T to A. Here on the other side of the plasmid, there's our other cut, the other side of the same single cut, AATT, AATT base pairing, and our insert from human DNA has been stuck into the plasmid. We have actually changed the plasmid. We've changed its sequence, and we have created a recombinant plasmid. Here you can see rDNA refers to the fact that this is a piece of recombinant DNA. And there we are. There's our plasmid with its insert. And now what we need to do, of course, is get that plasmid into the bacterial system again, into living bacteria, so that they can grow up, grow up, grow up, grow more, grow more, grow more, grow more. And all the time, they hopefully will be reading off and translating all the information that is contained here to produce lots of growth hormone for us. That's it at its simplest. Of course, if you're interested, there's way more information that is available to you. Okay, so that's that for the moment. There's one important technique that I want you to know about. And this is because this technique is not only used in bioengineering, in forming recombinant DNA, and although it is, but because it's also used in so many other, it has so many other applications. And this is called the polymerase chain reaction. This is probably most famous or most familiar to you from its use in forensics. And because what polymerase chain reaction does is it will take very, very, very small amounts of DNA and it will amplify them. It will replicate them over and over and over and over again until they reach a concentration which is large enough for us to measure or to do things with, to manipulate whatever we want to do. There is something of a limit. A polymerase chain reaction is limited to several thousand. It can do thousands of bases lengths, but not more than that. There is a limit to the size of DNA that can replicate. But there are techniques. If you want to replicate a very large piece of DNA, there are ways of breaking it up, doing smaller bits and then joining them together at the end. But for most applications, it is applied to relatively short lengths of DNA. It is extremely valuable in bioengineering, but it's also very valuable in forensics, in detecting disease. For example, both COVID, HIV, these are things which polymerase chain reaction is used to examine human body fluids, for example, and see whether there is pathogenic DNA in case of COVID, and HIV, it's actually RNA, but we have ways of doing it by polymerase chain reaction nonetheless. Okay, so it's in theory, a relatively simple technique, and it depends upon some properties of DNA. The first property of DNA is very easily melted. Now, what we mean by saying that DNA is melted is that you remember that DNA has this very strong backbone of phosphate and sugar, phosphate, sugar, phosphate, sugar. And the bases, the nucleotides, have bases which face to inwards. And those are what base pair, A to T, C to G. But those base pairings which stabilize the molecule and keep it in its lovely helical shape, those base pairs are easily separated by heat. We don't have to heat the DNA up very much before those base pairs actually separate from one another and the two strands come apart. Now, remember, where else did the DNA strands come apart? They came apart during replication, but that separation was by enzymatic action. Here, we're just using the mechanics of heat, and we're actually melting the DNA. But the most amazing thing about it is, if we raise the temperature and separate those strands, we can mix them up as much as we like, you can swirl them around, whatever. When you lower the temperature, they will come back with absolute fidelity. We will end up with exactly the same base pairing as original. It's a remarkable property of DNA, and it relies on the fact that although those base pairing bonds, which are hydrogen bonds, are very weak, there are thousands upon thousands upon thousands of them. And the only way that the DNA can satisfy all the requirements of bonding together is to come back in exactly the same base pairing sequence as it had before. So that's property number one. We can separate it and we can get it to come together again with fidelity. Now, when you think about what we need for DNA replication, what did we need? First of all, before we could replicate the DNA strands, we had to have a primer. We had to have an RNA primer, a short piece of RNA which base paired to the DNA and temporarily formed a hybrid DNA RNA strand. Once that primer was inserted, then replication could take place. And that replication was done by a DNA polymerase. And what it did was it pulled in nucleotides that were in solution and put them into order by looking at the existing strand and base pairing so that it added in the matching strand. So the polymerase chain reaction is an artificial replication system. So first of all, we have our DNA, our target DNA that we want to amplify. We want to get the concentration high. So the one thing we need to know is we need to know the sequence of that piece of DNA, because we want to know on either side, remember it's a double strand, for each strand that we're going to replicate, we want to know a short sequence on either side of our target DNA to which we can attach RNA primers. And once we know the sequence on either side of our target DNA, we can actually construct the primers. It's very easy to do, to synthesize short pieces of RNA, which we'll use as primers. So here are the things that we need. First of all, here's our target DNA. Second of all, we have primers, one to fit on one strand, the other to fit on the other strand. They are not going to be the same. Second of all, we need an abundant store of nucleotides because we're going to replicate this target DNA strand over and over again. And of course, the last thing that we need is polymerase. Now, here is a problem, and that is we're actually going to raise the temperature up to about 95. And that is way, way, way above the temperature for most proteins can operate satisfactorily. At that higher temperature, most proteins in enzymes would denature. They would be changed by they would be damaged by the heat so severely that they would no longer function. So this DNA polymerase that we're going to use is really special. It is extracted from a bacterium called Thermophilus aquaticus, Thermus aquaticus, sometimes you will see it called. And it is, so from that name, the TAQ, it's called Taq polymerase. And that organism lives in hot springs. So it is adapted to very, very hot water. and we can extract its DNA polymerase and use it because that DNA polymerase is adapted to high temperatures. Okay, so here's our mixture: target DNA, primers, and then the nucleotides. And with the addition of a DNA polymerase, hopefully we can get this whole thing to start replicating. The way we do it is this. We're going to take our DNA, and here's where we raise it. We incubate at about 9, did I say 95? They say 94 here. It doesn't much matter. To the mid-90s. We raise the temperature, and what happens when we do that is these two strands of the DNA are going to separate from one another. We now lower the temperature to about 60 degrees. That is hot enough that the two strands will not come together yet. They will stay apart from one another, but it is cool enough for the RNA primers to stick, to base pair to the beginning of the piece of DNA that we want to replicate. Here's the primer adhering to the one strand. Here's the primer adhering to the other strand. Now we raise the temperature slightly from 60 degrees. We raise it to 72 degrees. And at 72 degrees, the DNA polymerase will start operating, and it will replicate each of these strands. So what do we end up with? At the end of that process, what we end up with is 2 copies of our target DNA. What's really important? What is really important about this is we can specify which piece of DNA we want replicated by designing our primers. Our primers are the markers. Or rather, the primers recognize markers on either side of the piece of DNA that we are interested in. And only that piece of DNA is going to be replicated, not the whole genome. Okay, only that piece of DNA is going to be replicated. And we've ended up with 2 copies after one round of this sequence. We're now going to repeat the sequence. We're going to heat up again to 94, 95 degrees. We're going to melt our DNA again. It's going to separate. We're going to lower the temperature to 60, and the primers are going to stick back on to the piece of DNA. We're now going to raise the temperature to 72 again, and we're going to replicate again. And what do we end up with? We end up with 4 copies of our DNA. And we can do this over and over again. There is a limit, however. We can do it up to about 30 times. But as we go on, you must realize if any mistake is made here, that mistake is going to be replicated over and over again. So as we increase the number of repetitions of this, so we run the risk of incorporating error. So there's a limit, and it's usually between 20 and 30 replications. But in that, we end up with billions of copies of the target DNA, because each round, all the DNA that we have is replicated all over again, and very, very rapidly. We end up with high concentrations of DNA. So here's a little movie. I don't know if this is going to work on my, let's see.
The first step of PCR is denaturation. During this step, the DNA is incubated at a high temperature, which causes the DNA strands to separate by breaking the hydrogen bonds between each base pair. The second step of PCR is priming. During this step, the DNA is incubated at a relatively low...
Oh, shit, sorry. Oh, excuse my language. Oh, sorry. I'll do it again.
The first step of PCR is denaturation. During this step, the DNA is incubated at a high temperature, which causes the DNA strands to separate by breaking the hydrogen bonds between each base pair. The second step of PCR is priming. During this step, the DNA is incubated at a relatively low temperature, allowing the primers to attach to the single-stranded target DNA. The third step of PCR is extension. During this step, the DNA is incubated at an intermediate temperature at which the DNA polymerase rapidly replicates the DNA. During PCR, the temperature is cycled between a high temperature, a relatively low temperature, and an intermediate temperature. The high temperature denatures the DNA. The low temperature allows DNA primers to bind, and the intermediate temperature allows for the rapid replication of DNA. A machine called a thermocycler cycles through these temperatures repeatedly, doubling the amount of DNA present with each cycle. With each cycle through the polymerase chain reaction, the amount of DNA present doubles. After 30 rounds of PCR, the target DNA has been amplified by more than 1 billion times.
Okay. Let me see if I can escape this. All right. So that's the basic principles of PCR. Make sure that you understand it. You know you're going to get a question on your tests and exams, all right? So the very last thing, I'm sorry that I've ended up without time to go over this in class because this is such an incredibly important section. But I have extracted these two short movies for you to watch on this very latest set of biotechnology techniques, which depend upon a bacterial system called CRISPR-Cas9 to create a gene editing system. It's relatively complicated, I'm afraid. This is why I've not wanted to go into it in huge amount of detail. But I want you to at least know the very basic principles. Again, it depends upon a system which bacteria use to deal with viruses. It is part of their kind of immune system, if you want to think of it that way. And it consists of a pair of molecular scissors again, but this time molecular scissors which are guided to their target by a short piece of RNA which would match to a DNA sequence on the viral DNA. So the scissors are matched to a specific sequence. The CRISPR-Cas9 system uses that ability to cut DNA, but it manipulates it. It manipulates it so that where the cut occurs, we can either take out a base, and we can change base sequence, or we can insert a piece of DNA. This means that in living cells, we can actually use this CRISPR-Cas9 system to edit existing genes, not by inserting, we don't change by inserting a plasmid or whatever. We actually go in to the host DNA and alter it. And this is an unbelievably powerful technique. And it carries with it an enormous moral weight as well, because it offers us the chance to change a genome. And so we can actually start to think about changing a human genome, especially if we start to use CRISPR-Cas9 to edit things like gametes, for example, or trying to go into a fetus or design. This raises the specter of designer babies, for example. However, CRISPR-Cas9 has this huge potential, and that is to be used to treat genetic diseases. And in fact, this very week, they announced a huge advance in medical science. and that is the treatment, the very first treatment of this terrible disease called Huntington's chorea or Huntington's syndrome, Huntington's disease, which is a neurodegenerative disease. It's inherited, and if people have the gene, it inevitably leads to dementia and death, and sometimes at a very young age. But using CRISPR-Cas9, they have just announced the first cases where they have not cured the disease, but they have shown that they can massively relieve the symptoms of the disease and prolong life. And if, in fact, and the hope is that if, in fact, they can identify people very early on as children, for example, when they are carrying the gene for Huntington's disease, they will be able to alter the genes sufficiently that the child will never suffer from the disease. So this is a huge, huge advance. Please watch these. Please watch these two videos carefully. This is a technique which you are going to hear as nurses or in wherever you're involved in health sciences. You are going to hear about this over and over again over the next coming years. Okay, so I'm sorry that this whole section on genetic engineering and everything is rather rushed and rather superficial, but at least it gives you an inroad into the topics. Oh, by the way, sorry, this is a link here to an article which gives you the details of this. All right, so what does DNA actually do? DNA carries in its sequence the recipe for all of the proteins which the body needs. Now, the body consists largely of protein, various other biomolecules as well, but protein is actually structures the formation of our cells and consists of most of our biomass. So, number one, DNA is coding for all of our structural, all of our biomass and how it is put together. And second of all, most of our enzymes, 99.9% of our enzyme activity is by proteins, and those enzymes are coded for in the DNA. So DNA is driving all of the processes of the body. They're driving its building, and they're driving all of the chemical reactions, because all of our chemical reactions are governed by enzymes. So that is largely what this next section is going to be on. It's going to be how the body actually deals with two things, with energy and with matter. But all the way along, remember that all of the control actually lies in DNA sequence. So what are the characteristics of living things? What is it that makes something alive? What is it that we recognize when we look at something and say, that is alive? The first thing is that all living things grow and they all reproduce. They all build biomass. And when they reach a certain stage, they will reproduce. Even our own cells, for example, do this just as a bacterium does it. A bacterium grows to a certain size, then divides, it reproduces. Our own cells are the same. Our own cells grow to a certain size under control and then divide. Second of all living things have a metabolism. And we'll talk about a little bit about what metabolism actually is. I've already said it's something to do with matter, and it's something to do with energy. And metabolism is the management of matter and of energy in such a way that living processes can continue. So when we look at the metabolism, we're going to see some very complicated processes, but fundamentally the cell is directing the use of energy so that it can manage and control the growth and the manipulation of biomolecules. It's something to do with matter and with energy. The third thing is all living things evolve. If they didn't, we'd all still be prokaryotes swimming around in primordial seas. But all living things actually evolve. And that is characteristic of life, fortunately. Fourth, all living things are able to respond to stimuli. They're all able to differentiate function so that they respond to outward stimuli. And that is largely the responsibility of, again, of proteins, of specialized proteins whose recipe lies in DNA. The last thing is that all living things, to one extent or another, produce heritable information. They have to, because the properties of the cell must be passed on to the progeny. In the case of advanced cells, the eukaryote cells, this is sometimes in the form of genetic exchange, in the form of sexual reproduction. But even in prokaryotes, we've already seen there is a degree of genetic exchange. But all living things produce heritable information. They essentially produce a copy of all of their genetic information to pass on to their progeny. That is not always that accurate. That is why evolution is actually able to occur. So when we look at what metabolism is, or whether it's microbial or whatever, what we see is that it is the sum of all of the chemical reactions that take place in the cell. So it's the buildup and breakdown of nutrients, for example. It's the chemical reactions which provide energy and which create substances that sustain life. So again, here we come back to it. It is something to do with managing biomolecules to a particular purpose, taking those biomolecules either to build the cell itself or to break down biomolecules to obtain energy, to drive the processes of the cell. All of our metabolic processes are inaccurate. They are all inefficient. They all, every time metabolic reaction occurs, there is waste. And if you don't believe me, just put your hand on your arm and see what do you feel. You feel heat coming off. That heat, although we manage it very carefully as homeotherms, we manage our body heat. Nonetheless, the origin of that heat is from the inefficiency of our metabolic reactions. So we are continually losing energy. We have to gain that energy from somewhere, and we gain it from fuels. But at the same time, we have to build our bodies. We have to maintain structures. We have to repair structures. So our nutrients that we take in form two functions. They provide us with energy, but they provide us also with building blocks. And it is the responsibility of the metabolism to balance those two processes. Now, we are going to be talking about specific metabolic processes, but always remember that all metabolic processes are intimately linked to one another. And although we discuss it as if, for example, one set of metabolic reactions is a clean run through different reactions, remember that the products of reactions may actually be bled off into other metabolic pathways. Sometimes what looks like a metabolic pathway with a single aim is actually a metabolic pathway which is providing something to many different systems in the cell. Okay, so let's break it down to its most simple. There are two basic types of reaction that are taking place in living systems. And the first is that there are reactions of synthesis, of building, of taking simpler components and putting them together to make a more complex component. And these are the processes which we refer to as being anabolic. The anabolic reactions, they are synthetic reactions. But the characteristic of these anabolic reactions is that they require an input of energy. Because they require an input of energy, we say they are endergonic. That is the chemical term for it. And that synthesis is most frequently by taking simpler units. We already saw it in talking about nucleic acids joining together to, nucleotides joining together to form nucleic acids, or amino acids joining together to form proteins. And when they do so, we join them together by removing a water molecule. They are dehydration reactions. And they are, remember, they require energy. That is, they are endergonic. They are usually by dehydration reactions, and they are synthetic reactions. They are making something. The opposite of that are the catabolic reactions. And this is how we obtain the energy to do this, to do all of the work of the anabolic reactions. The catabolic reactions are reactions of decomposition, and they produce energy. They are exergonic reactions. They produce energy, and we can harvest that energy, at least some of it. We can harvest some of that energy, and we can make it into useful energy. And we put that useful energy into anabolic reactions to synthesize. So the catabolic reactions are reactions of decomposition, but the decomposition, of course, is directed, and it is by hydrolysis. We put back into molecules the water molecule we took out that was taken out somewhere in a dehydration synthesis. We put back the water molecule. That is called hydrolysis, and that splits the molecule into smaller components with the release of energy. So those two sets of reactions, the anabolic and the catabolic reactions, they fit in with one another to provide us with our entire metabolism. So catabolism, we take complex molecules and we break them down into smaller components and we extract energy from them. Anabolic reactions, we take simple molecules and with the input of energy, we make more complex molecules. Now, here's the energy that we talk about, where the energy that is traded back and forth does not fly around the cell like little lightning bolts. It is trafficked in very, very specific ways. And in almost all biological reactions, useful biological energy is brought in the form of this molecule called ATP, which we're going to be hearing a lot about. And the molecule ATP is actually a nucleotide. It's adenosine, but it has attached to it 3 phosphate groups. And the third phosphate group that is part of ATP is attached with a very, very energetic bond, which is very, very unstable. ATP is actually not a long-term molecule. It is relatively short-lived, but when it loses that phosphate group, which it does in the cell, loses that phosphate group under very controlled circumstances, the phosphate is removed together with its energy. And the cell uses that energy, and it's going to use that energy in anabolism, in building reactions. And the cell requires a constant supply of energy. We waste energy continually because of the inefficiency of reactions. We require continual input of energy in order to keep all of the metabolic reactions going. That is what the story of microbial metabolism is about. Where does that energy come from, and how is it that the cell manages it? Okay, so this is just to belabor the point a little bit. Okay, Here are the catabolic reactions. We take macromolecules, complex macromolecules. Let's think of fuel molecules, for example, oils, sugars, this sort of thing. And we're going to break those down into simpler molecules, which release energy. Anabolic reactions, on the other hand, require energy and simpler molecules. We take simpler molecules and we build a macromolecule. We build that macromolecule for a reason, because we need protein, because we need proteins that sense light, because we need proteins because we are growing a cell to the point where it is going to divide. We need an input of energy. So catabolic releases energy. Anabolic requires energy. And it is ATP that trades that energy back and forth. We need energy. We'll break down fuel. We'll grab the energy from the fuel, store it in ATP, and feed it into the anabolic pathways through ATP to do our building, to do the building of the cell. Now, the way in which the energy actually is traded back and forth is very specific. The energy, as I said, it doesn't just fly around. It doesn't go from a fuel like a little lightning bolt to ATP. In actual fact, the energy in the cell is traded in the form of electrons. And electrons are subatomic particles, obviously, that are in orbit around a nucleus of an atom. The surprising thing about the electrons is they contain an enormous amount of energy, an enormous amount of potential energy, that if you treat them right, you can actually harvest that energy. So the electrons are the coins. They are the nickels and dimes of the energy system. And the way in which electrons are exchanged between atoms is very important. If I have an atom and I take one of its electrons and I remove that electron, that is called an oxidation reaction. Now, if I take off an electron like this, I cannot have the electron just floating around willy-nilly. The electron has to go somewhere, and it is donated to another atom. So the loss of the electron is oxidation. And here is the oxidized form of A. So this is an ion. It has lost an electron. Its charge has changed in actual fact. Electrons are negatively charged. If I take away an electron from an atom, that atom becomes positively charged. So here is the positively charged ion, the oxidized form of A. But that electron has to go somewhere. And the electron goes to this substance, in this case, it goes to B. And B, because it gains an electron, now has a negative charge, but it has gained that electron. So this is also an ion. And this is actually called an anion. It's a negatively charged ion. This is a cation because it is positively charged. So those reactions, oxidation and reduction, they always go in pairs. You never have an oxidation reaction that doesn't reduce something, right? You never have a reduction reaction that didn't gain its electrons from oxidation somewhere. They run in pairs, in couplets like that. In biological systems, it is the electrons of hydrogen which are particularly important. Now, hydrogen consists only of two things. It has a proton, a positively charged subatomic particle in its nucleus, and it then has an electron, which is negatively charged. That electron is very easily removed from hydrogen. It can very easily be moved from hydrogen to something else to reduce it, leaving behind a positive proton. Now, usually we deal with the two together. The proton and the electron very often go together into a set of reactions. Sometimes the proton can be put into solution. And if I put a lot of protons into solution, then what I do is I create an acid. That is almost the definition of an acid. A, a, an acid is a solution which has an excess of protons of, of H, and that is H plus. Just put that into your brain and keep it there safe, along with 5' hydroxyl and all of the other things I've told you you're never allowed to forget. Put into your brain that a solution of protons creates an acid, an acidic environment. So what we're going to watch most closely now is this trade of electrons back and forth in the cell, usually derived from hydrogen. And here's why we are so interested in hydrogen. These here are both sugars here and fats here. You can see why they are such a good source of energy. They're a good source of energy because they have so many hydrogens associated with them. And those hydrogens are very easily abstracted. These are very stable compounds, by the way. They don't spontaneously break down. But so they form very good long-term storage of energy. But under the right circumstances, if we need energy, we can break down our fuels, either fats and oils, or the glucose and fructose and other sugars. And why? Because look at all of this. All of these hydrogens, all of them are able to provide electrons and protons to the cell. And it's particularly the electrons that we're interested in because of their enormous energy. So sugar is exactly the same. They're full of hydrogens, which the cell can use under the right circumstances. So that is that trafficking of energy that we're going to be looking at particularly closely. But before we start, we also need to understand a couple of things about metabolic pathways. So these complex reactions of the metabolism do not occur in isolation. So we very seldom see a simple path from one substance to another in metabolic systems. Instead, there are usually a set of intermediary steps in between. So this begins to look very, very complicated. But there's something about biological systems that you must always remember. And that is with complexity comes control. The more complex the system is, the easier it is in actual fact to control, because you can control individual steps. And by simply controlling individual simple steps, you can control the whole pathway. You'll see a good example right now. So here is a metabolic system. What do we want to do? We're going to start off with initial reactants, and we're going to end up with a final product, A to E. So why not just go from A straight to E? Why not just have a system which like breaks this down, right? Let's say that, and end up with this. Well, in actual fact, biological systems will use all of these intermediaries. We will have a set of intermediate reactions with intermediate products. So here's our initial reactant. This is an intermediate product. It's the first intermediate product. That is then put into a set of reactions to end up with a second intermediate, then a third, and then our final. Why do this? Because complexity offers control. And at each step, at each reaction here is governed by its own enzyme. So here's enzyme 1, enzyme 2, enzyme 3, enzyme 4. If I can control these enzymes, I can control a whole series of reactions. And I can end up with fine-tuning this reaction so that I fine-tune the amount of the product that I end up with at the end. this is often very, very important to the cell. The cell does not want to waste energy producing product it doesn't need. So the second advantage, each of these intermediate steps, each of these products is usually useful somewhere else. It usually can be abstracted to go into into a different pathway, to serve as the basis for another pathway, or to produce another set of products from the same initial reactant. So the important things, there are a set of intermediate steps, there are a set of intermediate products, and each intermediate step is governed by its own enzyme. And what we're going to see is that we can easily control the rate of this entire pathway by controlling enzyme activity. Okay, so that's the first thing. We're going to talk about what enzymes are and how they function, everything else in detail in a while. So the second thing, I showed you originally the sugars and the fats with all of their hydrogens. And immediately you see those hydrogens, you need to think about the electrons that are contained in those hydrogens. They are full of energy. They're incredibly rich energy source to the cell. But the cell has to have ways of actually grabbing that energy and putting it into useful form. And the ultimate useful form of that the biological systems use is that of ATP. And here it is here again, I've already mentioned it, is adenosine. Here's the ATP itself, adenosine triphosphate, adenosine plus 1, 2, 3 phosphates. Adenosine triphosphate is produced by taking adenosine diphosphate, adenosine 1, 2 diphosphate, adding energy and another phosphate to produce adenosine triphosphate. It is this last bond here which is of particular energetic importance. There's a lot of energy here. And this bond is actually unstable. Because it is unstable, ATP is actually a short-lived energy transport mechanism. It is brought in immediately. A set of reactions require it because it will soon break down and release this phosphate. But the ATP, you can think of it like the purse. I said that the electrons were the coinage. ATP is like the purse carrying that coinage. And you open the purse and you give out the coinage when you need to. So I would say very occasionally the cell may use this bond as well, but it's not that common. But just know that it is possible to use this bond if circumstances of a particular metabolic reaction demand it. Now, the way in which the electron and all of its energy are transferred is usually by taking the whole phosphate, not just the electrons, but by taking the whole phosphate and putting it onto another compound. And that compound is called an electron carrier. We can't just have electrons flying around the cell, even over short distance or a very short time period. Instead, electrons are actually transported by electron carriers. And those electron carriers are usually phosphorylated together with electrons from adenosine triphosphate with all of their energy. So in summary, here's what we have to deal with so far. Here's our ATP, adenosine triphosphate. This is a molecular model of it. Adenosine triphosphate serves to traffic energy between catabolic reactions and anabolic reactions. So the most common catabolic reactions that we are going to encounter are those of energy breakdown, of energetic compound breakdown, like oils, fats, etc., sugars. They get broken down and energy is released. Here's the example of glucose. Glucose can be broken down to simple compounds, to carbon dioxide and water, with the release of a lot of energy. And that energy from glucose breakdown is stored very temporarily in ATP. That energy stored in ATP can be used to take an oxidized compound, like the amino acids. We're going to take simple components, the amino acids. They are already oxidized. And we are going to reduce them. We're going to reduce them by taking the energy, the electrons contained in this ATP and all of the energy. We're going to reduce them. And in doing so, we're going to push them through anabolic reactions to produce proteins, for example. So we're going to, if we want to take oxidized compounds and we want to change them to highly reduced compounds, in other words, with lots of electrons derived from hydrogen, then we're going to use ATP as the source of those electrons and that energy. That these compounds here, by the way, maybe storage compounds, whatever. We may actually later break them down. We may break them down again in catabolic reactions to extract more energy. But the cycling between catabolism and anabolism is the energy transfer is always mediated by ATP. Unfortunately, of course, ATP can't be stored. It's really very unstable. It's a short-term trading mechanism. And if we want to store energy for any length of time, it has to go into long-term storage compounds again. And it just needs to be mentioned. In eukaryotes, those long-term storage components are usually either carbohydrates, such as starch and glycogen, and that's the other compound in eukaryotes, or in the lipids and fats. So as I showed you originally, both the sugars, starch, glycogen, these are polymers made of sugar units. They contain abundant hydrogen together with their electrons. That is why they are storage units, but they are very stable storage units. They do not break down spontaneously. Instead, if we want to break them down, we're going to have to use enzymatic systems in order to do so, but that offers us control over the energy and allows us to put that energy into biologically useful form in the form of ATP. In the prokaryotes, the long-term storage systems are to build up glycogen and also other substances like this is called polybeta hydroxybutyrate. That's a very common storage compound, and I mention it because it is sometimes visible in bacterial cells as little clusters of cellular inclusions. So these are all long-term storage, very stable. And if we want to access the energy, we have to use enzymatic systems to do so. And that is going to be the subject of considerable amount of our discussion. Before we go any further, I know that people have not done much chemistry. So I just want to give you a basic primer on how chemical reactions are actually driven, how and why they occur. So a fundamental understanding about chemical reactions is that if I want one type of molecule to react with another molecule, two things are required. First of all, they must be close to one another. Second of all, when they encounter one another, they must do so with sufficient force that they disrupt and force a reconstruction of the chemical bonds. So what we're going to watch happening is a restructuring of the molecules because they smack together so hard. And this is called the collision theory. Now, so another fundamental principle of chemistry is that with increased heat, the kinetic energy of molecules increases, right? So increased heat, molecules just move faster. And if they smack into one another, they smack into one another harder, and it is more likely then that a chemical reaction will occur. So let's just have a look at a simple set of reactions. This here is methane, and that is common household gas, and methane is produced by prokaryote systems. You'll remember I told you at the very beginning of this course that the archaea are responsible for the production of much of the Earth's methane. We use it as household gas. And it contained in it is an enormous amount of energy because it has, for every carbon, it has 4 hydrogens attached to it. So let's say we want to use this household gas to produce energy in the form of heat. That's what happens on your gas stove. Or we're going to burn it with oxygen. But if I mix methane and oxygen together and I just let them mix and I do nothing else, I leave them at room temperature to mix, every now and again in that mixture, 1 or 2 of the molecules, methane and oxygen, might smack into one another sufficiently that they will alter their structure and end up breaking up into carbon dioxide and water. So that is one thing. These reactions take place spontaneously if there is sufficient energy to drive them. If I start to heat this mixture of methane and oxygen up, I increase the kinetic energy. And if I increase the kinetic energy, I increase the rate at which they encounter one another, and I increase the speed at which they collide with one another. If I do this sufficiently high, then they will start to spontaneously carry on the reactions, because every time this reaction occurs, they release some energy. And so when I get to a point where they release enough energy that energy itself can drive the reaction in other molecules, then the reaction will proceed spontaneously. That's when you see the flame light up. But before that flame lit up, some of these reactions were still occurring. It's just that we have to create a situation where sufficient collisions occur at sufficient kinetic speed that the whole reaction proceeds spontaneously. So that amount of energy that I need to put in to get the reaction to carry on its own is called the energy of activation. And in biological systems, I should tell you, the energy of activation for most biological reactions is very, very, very high. It's far above what the cell itself can produce. So just for the moment, just keep that in your mind. Most biological reactions do not occur spontaneously. So if we want biological reactions to occur, we have to intervene. And there are various ways we can intervene. We could increase the temperature, certainly for in organisms like us that harvest our heat, harvest the wasted energy of metabolism. We do raise the rates of our reactions by doing so. The second way that we can increase the rate of reaction is to actually increase the concentration of the substrates. And so if I increase the rate of concentration of methane and oxygen in a vessel, for example, then the more methane, the more the oxygen molecules there are, the more likely they are to encounter one another. But the way in which biological systems deal with this requirement is that they use a system of compounds called enzymes. Now, most of the enzymes are in fact proteins. But I say most because I've already mentioned one example where this was not the case, where enzymatic activity was done by something other than a protein, and that is by RNA. And you'll remember the ribosomal RNA had enzymatic activity. There are various RNA molecules in the cell which have enzymatic activity, and we call them the ribozymes. So we need to think now about what it is about enzymes that makes them so important in the propagation and the control of biological reactions. So here's an example of a reaction. We have reactants. It's a compound, AB, and we want to actually, let's say that this is a fuel compound, and we want to split it up. We want to drive it through a catabolic reaction, separate the two, and extract some energy. Well, if we just have this compound sitting around, we ask the question, does this compound actually react like that? Does it ever break down spontaneously? If I have a bowl of sugar and I set it on the table, that is an excellent source of energy. It's full of hydrogen. How many of those molecules break down spontaneously? The answer is not zero. There are, every now and again, maybe one molecule gets hit by a cosmic ray, something like that. Maybe just some extraneous source of energy enough to break up one molecule occurs or drive some of the hydrogens off or whatever. These reactions do occur spontaneously, but they occur very, very, very slowly. Very, very slowly. And the reason for that is they have a very high energy of activation. We need a lot of energy input to get them to actually react spontaneously. So this is the reaction without an enzyme. This is what an enzyme does. It actually lowers the energy of activation. And it lowers it so much that the probability of the reaction occurring is vastly increased. This is what enzymes do. They are catalysts. They are biological catalysts. And a catalyst is something that facilitates a reaction, but is unaffected by the reaction. It emerges, it participates in the reaction, but emerges unchanged from the reaction afterwards. We'll see this in numerous examples. So the enzymes are going to interact with the reactants, and they're going to end up facilitating the production of a product. And they do so by lowering the energy of activation that is required for those chemical reactions to occur. So this is the way in which enzymes actually operate. They operate by temporarily interacting with the substrates to form a complex. So here's our enzyme here. The enzyme has a very specific area where it will bind the substrates. And once it's bound the substrates, then can proceed to facilitate the reaction. In this case, what we're looking at, we're going to look at sucrose, as I mentioned before, very, very stable molecule. Very seldom breaks down spontaneously. Certainly a bowl of sugar sitting on the tabletop is not going to spontaneously burst into flames to produce carbon dioxide and water or anything else. But this is an enzyme which is able to interact with sucrose. And to do so, it has this specific site which binds sucrose and is specific to the shape of sucrose. And that is called the active site. Enzymes are very, very specific for their substrates, extremely specific. And is usually determined by the shape and by various other chemical properties of the substrate. In this case, it binds sucrose. The sucrose consists of two molecules of fructose and glucose joined together. And this enzyme is going to actually split those into one from the other. In order to do so, it binds it very tightly, but temporarily, into what is called an enzyme substrate complex. And once that occurs, then the enzyme facilitates the breakup of the molecule. Now, in this case, what it does is it actually physically does it. The enzyme binds the sucrose, and when it binds the sucrose, the enzyme itself changes shape. And it changes shape sufficiently that it stretches and breaks that bond between the glucose and the fructose. It releases the products, glucose, fructose molecule, because once that happens, they no longer fit the active site. And so they are released, and the enzyme emerges unchanged at the end of it, ready to go through the whole process again. These are the products of that reaction. The substrate is sucrose. The enzyme acts as a catalyst, and it does so by forming an enzyme substrate complex. And once that enzyme substrate complex is formed, then various things can happen with that enzyme. It drives processes in the enzyme. Either the enzyme flexes, or the enzyme adds groups, or the enzyme changes a chemical property. All sorts of things can be done by the enzyme, but the end shot is that it vastly lowers the energy of activation, and it repeats the process over and over so rapidly that we actually start to produce product far more rapidly than if we just left the substances alone to react on their own. Okay, so anyway, this is another enzyme system, but this one here is called invertase here. And here's just another way to represent the same reaction. Here's sucrose, and here is its glucose molecule there. Here's its fructose molecule. And the invertase, which is this enzyme here, ends up stressing that bond, this bond here, so much that it breaks and produces the glucose and fructose. So get it straight in your mind. What are the substrates? What are the products? And which is the enzyme? Especially when it's represented like this, not in that nice graphic form that we saw before. All right, so I'm going to end it there, and we will continue with this, of course, when I see you on Monday.